So now we are going to begin the chapter 3, talking about the nucleotide, like evolutionary changes in nucleotide sequences. So in any given time, if a person is dealing with the DNA sequence data, so I think previous week we're starting to talk to you about the DNA sequence and what's the format in the computer, because now a lot of sequence you can obtain from the database. And so I'm sure you already know a little bit about the sequence information from the database. And some of you might be working on your thesis project, which involve in sampling DNA sequence or understand the DNA sequence polymorphism in the population. So you might have a little bit experience. But when we want to calculate the differences between the two sequences, so this is the chapter really starting from this point, then we can do a lot of things. So we can estimate the divergence between the two sequences. We can use that as a parameter to reconstruct the phylogeny of the given group of organisms. And we also can estimate the evolution rate of, for example, members in the same gene family. So we do a lot of things using this sequence information. And it's very important because that's something we start with. And actually, that's something acting on or operating in our genome all the time. Most of us use DNA as the genetic material. So when we talk about mutation, what we mean is a nucleotide change at the sequence level on the chromosome. So that's the mutation and mutation rate we are concerning about. So this is what we rely on to calculate the distance differences. So if there's sequence in your genome and my genome, and we can compare the sequence divergence between the two sequences. So this is a sort of a common practice in this field. And we also want to use those information to look at more sophisticated statistical analysis, like a Bayesian analysis you will be told in the later lectures. So I'm sure Professor Hu will mention about all those statistical models in his lecture and also in our laboratory practice. So how do we begin to estimate or calculate the differences? And that's the entire the focus of this chapter. So just think about a very simple method. A simple method is the proportion of differences, right? If I give you two sequences with ATGC on the sequence in a row, then you can do an alignment between the CHAU-TI sequence. Then you can count the differences one by one. Right? Look at the sequence. If they are all A, they are the same. If this one is an A, the other one is a C, then that's the differences. So it's a very simple calculation. You just look at the sequence alignment, then you calculate the differences. I want to add one thing about the alignment is once you align the two sequences, what does that mean? When you do alignment between the two sequences, what's the fundamental thinking you are doing? Anyone? Can anyone give an answer? Yes?
You mentioned that two sequences have some homology.
That's right. So it's an evolutionary thinking. No matter which field you are, when you align the two sequences, actually in your mind, the sequences are similar based on homology. That's really good. So you are assuming, even you didn't say, you are assuming the two alleles are from the common ancestor. Then they accumulate mutations along the evolutionary time. And at the current stage, we are comparing the differences between the two. So that's the evolutionary approach. So even people are in bioinformatics and genomics, they didn't really say based on the evolution, but when they align the sequence, that's the fundamental thinking we have in mind. Okay. So the simple calculation about the differences is the calculation is counting method. So I count the number of differences divided to the total length of the sequence I'm working on. And that's the proportion of the differences. And that's the P here. So the P, if I have 10 differences in the given 100 base pair region, then the proportion of the differences is 10 divided to 100, and that's 0.1. So it's 10% of the differences in the given sequence. So that's the simple calculation. However, that's the observed number. That means you have two sequences and what's the differences. But that doesn't tell you what's happening in the past, in the historical time. Because we're only dealing with the nucleotide sequence. And the nucleotide sequence is very simple. We only have 4 different choices. And for any given site, they can change back and forth. So I'm going to illustrate that in the following slide. So any given site could change to one nucleotide and change to a different one. But at given time point, you only look at the 2 status, which is just assuming we are only comparing 2 sequences. And this is what I'm going to illustrate here. So this is a sequence assuming, so on the top, so this is, I put on in the handout, and also this is from your textbook. On the top is the given ancestral sequence. I just put it in horizontal. And the vertical one is from the first position to the end. That is homologous to this ancestral sequence. Marking in red, that's the one which shows difference between the 2 sequences. Assuming we know the ancestral sequence, that's the case here. Then for any given one, like the one I put a box here. So for this side, both sequences are T, right? Both sequences showing the same nucleotide. When we align the 2 sequences, we'll say, okay, there's no difference at this position. But since I told you the ancestral sequence on the top actually is an A. So given this case, at least you will say, well, the 2 sequence at this has 1 mutation occur independently from A to T. But that is not observed when you just simply put the 2 sequences together. So that's what we want to say. We want to estimate all the changes during the evolutionary time. And in most cases, we actually don't know the ancestral sequence. I have said that many times in this class. A lot of practice we are doing is we get the sequence from the living organism at a given time. Then we want to estimate what happened in the past. So that's what we do. So this is just a case, try to explain the observed differences. It's not exactly the same as what happened during the past. And this is a simple, just using this example to show you we usually underestimate the changes.